internet issues, and so it's good that you found time that we could cover it today. I must have had a glance on how your teaching timetable is running, and soon I think you should be having some continuous assessment test done. Of course, that would be administered online, and thereafter we would have continuity of the lectures uh, running into the immune response mechanisms to infectious agents. I think not only infectious agents, but also the hypersensitivity reaction, which is coming just ahead. Now, why do I recount on those? Because the lecture of today is not solitarily, like I had mentioned to you sometimes earlier, that a number of these lectures, nearly all of them, are interlinked. So one builds onto the other. And therefore, the lecture of today on cell-mediated immunity is just running relative to what you could have learned also on humoral immune response, but then it builds onto a background to help you understand what is coming ahead on immune response to infectious agents, not only to infectious agents, but also immune response to tumors, and also in hypersensitivity reactions, particularly the hypersensitivity type 4 reaction, which predominantly require the cell-mediated immune response. So the in-depth of today, you should be able to know what aspect of immune response require cell-mediated immunity, and you have therefore to categorize these responses into particular cells which are triggered. So if we talk about humoral immune response, you'd be thinking largely the B cells which are activated. But when we come to cell-mediated immune response, you would be largely thinking about the T cell response. And T cell response, not only T cell response, but we also have activated macrophages. We'll see along how it goes, because by now you should be knowing that macrophages are cells which are predominantly important in innate immune response. Then it should not be categorized under cell-mediated immune response. But therefore, how does it cross to come into the cell-mediated immune response? We'll understand this shortly. So we do know that host defenses against extracellular infectious agents, and particularly the extracellular bacteria, can be defended against by the antibody secretion. By now, you should have also learned about complement activation, the classical and also the alternative pathway in particular. And we also have macrophages. I've just mentioned macrophages a short while ago, that these are very important at initial stages of the immune response, and particularly to the extracellular agents or infectious agents. But when we have intracellular infections, and these predominantly occur when you have got viral infection, or you also have intracellular bacteria like the TB antigen, they trigger what is called the cell-mediated immunity. So cell-mediated immunity will be responsible for these intracellular pathogens, and also to fungal and protozoan infections, because they are not most of the time considered the intracellular. They would be considered the extracellular per se, yes, but they cannot be phagocytosed. For instance, for example, you have got the Ascaris lumbricoides. You would be knowing it is so large to be phagocytosed. You have got, say, Plasmodium falciparum, which infects the red blood cells. So the red blood cells are enucleated, and therefore they cannot present this antigen to the effector T cells. Even if you talk about the fungal infection, take any typical example you want, and you'd note that they present difficulty in phagocytosis, and therefore the cell which would be called upon to clear out that cell which has already been infected by either fungal or protozoal infections are the T cells. So T cell will come here to kill the target cell. In this case, we talk about that cell which has been infected by fungal or protozoal infections. So largely, we look at it here that fungal infections and protozoal infections, they would not per se be considered intracellular pathogens, yet they require cell-mediated immune response because they present inherent problem of phagocytosis. So they cannot be phagocytosed, antigens broken down into smaller peptides, some which would be presented on the surface of the antigen-presenting cell. We also have the tumors, and tumors are considered intracellular antigens because they are generated from within the cell which has been transformed. You definitely know of the tumorigenesis, that a transformed cell generates new antigens. So these new antigens are built from within the cell which has been infected. Hence, we classify tumor antigens as intracellular antigens. So wholesomely, these are the elements which would trigger the cell-mediated immune response. So what does cell-mediated immune response do after it is activated? It plays a role in some harmful conditions, though, like in the hypersensitivity reactions type 4, we give example of contact dermatitis, but you also have tuberculous reaction when someone is infected with the TB bacteria, and we have the granuloma giant cell formation. So that giant cell formation would continuously trigger inflammation. So this is a prologue of the cell-mediated immune response. We also have the graft rejection and also the autoimmune diseases. These are all harmful conditions which are triggered when cell-mediated immune response is activated. However, we have got cell-mediated immune response, like I've mentioned in the previous slide, are important for clearing out the cells which are infected by, say, fungal or protozoal antigens. And we also have the intracellular bacteria as well as the tumor antigens. So the key cells which are important for this cell-mediated immunity include the cytotoxic T cells, otherwise termed the CD8 positive T cells. And you also have the natural killer cells. You know, natural killer cells are also lymphocytes and fall under the category of the cytotoxic T cells, the cytotoxic C cells, which can kill the pathogen. And we also have the activated macrophages. So previously, I mentioned to you that macrophages are key in phagocytosis, but in this scenario, these macrophages are macrophages which have to be triggered to be activated before they can carry out their tumoricidal effect or bactericidal effect in case they're responding to tumor or the intracellular bacteria respectively. And the characteristics of cell-mediated immunity, it must be subpopulation of T lymphocytes, which are important, like I mentioned to you, the natural killer cells and also the cytotoxic T lymphocytes. And we also have the macrophages and their products. When we talk about the products of macrophages, we largely refer to the cytokines which are produced by these macrophages, and these are able to clear out the pathogen in question, intracellular bacteria or viruses, or the tumor antigens when the body comes into contact with them. But when we continue the characteristics of cell-mediated immunity, we have got macrophages able to present the antigen through their surface receptors to the T cells. And these macrophages must express onto their surface a third molecule, the major histocompatibility complex molecule, which by now I think you've learned. I'm not sure if, yes, you should have learned major histocompatibility complex molecules, class 1, class 2, class 3, and their importance in not only determining whether a tissue can be rejected or accepted, but also their role in the T cell signaling or cell signaling, in which case they are utilized to present antigens to the T effector cells. So macrophages, once they recognize the antigen, they phagocytose it, they process it, and some of the processed antigens are expressed on the surface of these macrophages, bound on to major histocompatibility complex molecules. Then in that case, the T cells can be able to recognize it. So here, one thing you need to note that T cells are the ones which require the antigens to be presented to them bound to major histocompatibility complex molecules, not B cells. B cells are in themselves alone also antigen presenting cells. So once the antigen is presented, the T cells will recognize this antigen through their specific T cell receptors, and this will lead to clonal expansion, and the T cell will become activated and begins now to proliferate to make more copies of itself. Here, when it requires the proliferation and making more copies of itself, the key molecule which is important for triggering that kind of proliferation is called the cytokines, small regulatory molecules which are produced only when that response is required. I believe by now you should have tackled the topic cytokines, yeah, because it comes a bit much more earlier. But if you've not covered it, then it would be coming along. So cytokines are small regulatory molecules which will be able to affect the cell-mediated immune response, and also they're having key role in humoral immune response, which we are not going to tackle in this topic of today. We are going to concentrate largely on the cell-mediated immune response. So the role of cytokines is to help the proliferation of these, actually firstly activation and proliferation of the T cells, so that they make more clones of that T cell which has been activated. I'm not sure whether you have learned about, yes, I think it's I who must have taught you earlier time, T cell signaling and activation. So the molecules which are important for the T cell signaling and activation are the cytokines. So the key, the first primary thing which would happen is when the body comes into contact with a pathogen, there must be attraction to the site where the infectious agents has been recognized by monocytes and macrophages. So there is a chemotaxis of the monocytes and macrophages, and also even the lymphocyte to the site where the pathogen has intruded the body. Then the macrophages become activated. At that point, they can kill the pathogen directly. But when you go back, I mentioned to you that macrophages by themselves are antigen-presenting cells. So they will try to engulf that antigen and then phagocytose it, processes it, and then presents it to the effector T cell. However, recall that I mentioned to you that cell-mediated immunity is geared towards those antigens which are difficult to phagocytose. Typical, we have got the fungal and protozoan infection. So apart from the intercellular pathogens, which would, I'll give you a typical example with the HIV virus. The HIV virus, when it infects a cell, by the time it buds off to infect another cell, it ensures that the cell which was previously infected has been killed. So to circumvent this continuous death of the cells, the cell-mediated immunity, especially the activated macrophages and cytotoxic T lymphocytes, will target to kill that cell which initially got infected before the virus buds off and leaving the cell which it previously infected killed. So the cell-mediated immune response or cytotoxic T cells will target the virus-infected cell so that it kills it wholesomely without allowing it to support the viral replication and budding off to infect continuous cells. So the activated macrophages at the site where the pathogen has been recognized can be activated and kill that cell which is infected, yes, but that if defeated, we have got cytotoxic T lymphocyte, the CD8 positive T lymphocytes, which would now be able to directly kill the virus infected cells, tumor cells, and also the graft rejection. I mentioned to you that we also have the natural killer cell, which by themselves are also lymphocytes, and being activated, they are also able to be cytotoxic. I mentioned this in the previous slide, that they're also cytotoxic cells, which are able to kill the targeted cell directly. But cytokines, this is not the only thing that cytokines do, because I mentioned to you that cytokines also enable activation of the humoral immune response, which I don't want to talk about today, because you must have covered it before this lecture. So cytokines are also able to stimulate B cells to differentiate into plasma cells that secrete antibodies. And some of these antibodies, if they target virus, they can be neutralizing antibodies, which are able to neutralize the antibody, the virus, and inhibits their continuous infection of other cells. So let's look at phases of cell-mediated immunity. I think we have got three phases of cell-mediated immunity. One, which is the antigen processing and presentation, which I've largely talked about, mediated by the macrophages. So protein antigens processed and converted to peptides, they are bound to major histocompatibility complex on the surface of the antigen-presenting cells, and these then are presented to the T cells. Are we together so far?
Yes.
Or I'm slow, I need to speed up.
We're together.
Okay, so let's continue. So we are in the phases of cell-mediated immunity, which I've mentioned. The first phase is the antigen processing and presentation. I've explained to you. This is mediated by antigen presenting cells and presented to the effector T cells, the T cytotoxic lymphocyte. Remember that T helper cells are also antigen presenting cells. So the cell in question here, which we are talking about, can either be the helper T cell or the cytotoxic T cell, but we'll see the difference. Why do we talk about the helper T cell being antigen presenting cell? And on the other hand, we are talking about that antigens can be presented to them. Just shortly, it will come out clearer to you in terms of the major histocompatibility complex presentation restriction. So still on the antigen processing and presentation, we have got the extracellular proteins, and these are internalized into vesicular compartments of antigen presenting cells. And these largely would be the dendritic cells, macrophages, and B cells. I mentioned to you, B cells are also antigen presenting cells, and macrophages, and also dendritic cells. So this would be degraded to generate peptides, and some of these peptides would bind to major compatibility complex class 2. Now, I made a post there because I'm talking class 2. So when you talk about the major histocompatibility complex restriction of antigens, we have got extracellular proteins being presented to the T helper cells, the CD4 positive T cells, and they would only be presented when the antigen is bound to major histocompatibility complex class 2. Remember, we are talking largely that here we need cell-mediated immune response and not talk about much overtly on extracellular antigens, because we mentioned in initial slide that cell-mediated immunity is geared towards intracellular antigens and not extracellular antigens. However, when you talk about those antigens, say the tumor peptides which are expressed on the surface of the tumor cell, they would be recognized by still these antigen presenting cells and absorbed, and then can be broken down to stimulate the T helper cell. This does not end there, because T helper cells is not a killer cell. Remember, I mentioned to you only 3 cells which are important in cell-mediated immunity. We have got cytotoxic T cells, we have got natural killer cells, and we have got activated macrophages. So in the event of antigen processing and presentation as a phase of the cell-mediated immune response, how does it link with the end point of the killing of that target cell by the cells which are important in cell-mediated immune response? Then cytotoxic T lymphocytes, the natural killer cells, and the activated macrophage. So antigens which are recognized by the antigen presenting cells, be it tumor antigens which are presented on the surface or expressed on the surface of the tumor cell itself, recognized now by the dendritic cell, macrophages, or the B cells, they would capture, or they would pick these antigens and then process them and present them to the T helper cells. So this is the initial point of activation of those key cells which are important, and especially the cytotoxic cell. Once the CD4 T cell has been activated by the presentation of these antigens, they secrete cytokines. And their secretion of the cytokines is what will now follow the activation of the cytotoxic T lymphocytes. And when we talk about endogenous antigens, which now fall largely in what we were talking about in cell-mediated immune response, typical example is viral antigens, and also still the tumor antigens, or we talk about, say, those cells which are now frustrated in presentation of the antigen because they are infected with intracellular bacteria, like TB antigen. What will happen? We have got that cell still will try to process its antigen, and in a bid to present the antigen to the effector T cell, they will be required to express on their surface major histocompatibility complex class 1. So for endogenous antigens or intercellular pathogens, they require major histocompatibility complex class 1 to present the antigen to who? To cytotoxic T lymphocytes. On the other hand, the extracellular antigens or extracellular bacteria, for instance, it will be presented by antigen presenting cells, and the antigen must be bound to major histocompatibility complex class 2. Presented to who? to to the CD4 positive T cells or the T helper cells. So you make a distinction of this, that CD4 positive T cells equates to major histocompatibility complex class 2. Cytotoxic T lymphocytes equate to major histocompatibility complex class 1. So once the peptide major histocompatibility complex class 1 is expressed on the surface of the antigen presenting cells, they are recognized by the cytotoxic T cells or CD8 positive T cell, and then the CD8 positive T cells will kill through 3 modes using cytokines, granzymes, and perforins. I'll repeat there. The CD8 positive T cells do this killing of the target cell through 3 different modes. We have got the elaborated cytokines which they will produce, or perforins. Perforins are molecules which are able to cause pores on the surface of the target cells, or we have got granzymes. Granzymes are also enzymes which will typically catalytically break down the surface, the membrane surface of that target cell and penetrate into the target pathogen. Here in this case, we can talk about the viral antigen and kill that virus within the target cell. Then phase 2 of the cell-mediated immune response is the activation of the T cells. I mentioned to you that once the cells are stimulated, they get activated, and the last phase will be clonal proliferation to make more copies of each cell. So in this second phase of the T cell cell-mediated immune response is the mature anti cells being activated by 2 signals. And I remember it's me who was explaining to you that a student must have asked a question related to this or closer to this, but the subsequent lecture I came back and explained to you, or maybe I explained it through the email, that we have got 2 key signals during the T cell activation and antigen presentation. The first signal is the recognition of the antigenic peptide major histocompatibility complex on the surface of antigen presenting cell by T cell receptor complex. Of course, you know now by T cell receptor complex, CD4, CD3 is part and parcel of the T cell receptor. If you look at the T cell receptor complexity, CD3 is part and parcel of the T cell receptor complex. So what only differs is the core molecules which would be expressed on the surface of the T cell. So the core molecules can either be CD4 or CD8. Requirement of CD4 is that that antigen must be brought to it by the antigen presenting cell bound to major histocompatibility complex class 2. The requirement of the CD8 positive T cells is that for it to recognize that antigen must be brought to it by antigen presenting cell bound to major histocompatibility complex class 1. So this is the only difference. So that forms the first point of the signaling, which would enable now this T cell in later phase to clonally expand. Then the second signaling event in the activation of the T cells is the interaction of the other molecules, co-stimulatory molecules, in this case, particularly the CD28 on the T cell with CD7 on the antigen presenting cell. So these are co-stimulatory molecules which are required in the second phase of the T cell activation. I think this I must have also captured with you when we were talking about the antigen presentation and T cell signaling. Yes. Then the T helper cells will express cytokine, in this case interleukin-2, and this interleukin-2 will auto-activate these T helper cells. You remember this very well. I talked about it in the antigen presentation and T cell signaling. So the antigen presenting cells will release interleukin-1, and this interleukin-1 will auto-activate the antigen presenting cell to give a signal now to the T helper cell. So the T helper cell in itself again will further secrete interleukin-2 to auto-activate itself. So the elaborate cytokines which would be produced after the T helper 2 has activated the T helper cells are the ones which would be now important in activating the macrophages, natural killer cells, B cells, and as well as the cytotoxic T lymphocytes. So the sequel of events are, we have got interleukin 1. Interleukin 1 is to help the interaction between the antigen presenting cell, the antigen it is carrying, and the T helper cell. Then the T helper cell will produce interleukin 2, which will act in an autocrine way, activating it from the resting T cell to activated T cell. Then thereafter, the T helper cell will elaborate further cytokines, which would be important for now activating the macrophages, natural killer cells, B cells, as well as cytotoxic T lymphocytes. So yes, I've mentioned here the activated T cytotoxic lymphocytes, which is activated by the secretion of the cytokines by the T helper cell, which was auto-activated by the interleukin 2. So finally, then the cytotoxic T lymphocytes and also activated macrophages plus natural killer cells are the ones which would be important for targeting the cells which are carrying intercellular pathogens or the tumor antigens. And in this case, we talk about the antigens being expressed and presented to the cytotoxic T lymphocytes bound to major histocompatibility complex class 1. You will not forget about this, that cytotoxic T lymphocytes equate to MHC class 1. The T helper cells or CD4 positive T cells equates to major histocompatibility complex class 2, and that responds to now the extracellular bacteria, which we are not talking about here today. Then the last phase of the T cell-mediated immunity is finally the activation of macrophages and also the delayed type hypersensitivity, which occur as an event of the secretion of additional cytokines, in this case, the interferon gamma. And this interferon gamma will activate macrophages and increase their ability to kill the ingested intracellular pathogens. And I give you a typical example here, the TB antigen. So I told you that this is an adverse event reaction, like an autoimmune reaction, like also the hypersensitivity. So sometimes the cell-mediated immune response is important, is good, but it can also result into hypersensitivity reaction or overt damage because of the autoimmune response. So you need to look into the cell-mediated immune response as a benefit and also as a disadvantage in the body. And the process of activation of macrophages, natural killer cells, cytotoxic T cells, and also the infiltration and proliferation of inflammatory cells, all this will be stimulated by these cytokines, which are released by particularly the T helper cell, because we looked at it how it went. It went from the antigen presenting cell stimulating the T helper cell in an autocrine way activate itself from the resting T cell helper cell to the activated T helper cell. From there, it now can recognize antigen, which would be presented to the cytotoxic T lymphocytes. And the cytotoxic T lymphocytes, the activated macrophages, as well as the natural killer cells are the ones which will be key in clearing out the pathogen which we are talking about here in question. The intercellular bacteria, the tumor antigens, we talk about the fungal and protozoan infections, as well as the viral infections. And lastly, I think this is the last slide, that activated macrophages I've mentioned to you can be able now to kill the target cell. And the cytotoxicity is non-specific, and I've mentioned to you, just like the cytotoxic T lymphocytes, we have got also these macrophages using the cytokines. And here in the cytokines we talk about is the tumor necrosis factor, as well as the oxygen radicals, the nitric oxide enzymes, as well as oxygen metabolites, which it uses during this delayed type of hypersensitivity reaction. And in case the infection is not fully resolved, the activated macrophages will now cause tissue injury and fibrosis, which is witnessed in delayed type sensitivity reaction. So this is why we talk about this cell-mediated immune response may be beneficial, not even maybe, in large part it is beneficial to the host, but to an extent it can end up causing the fibrosis and injury to the tissue. And then you as a doctor, you are presented here with a complexity which you thought you were resolving, but now you have to treat a different condition altogether. So that is the summary of what we would learn about cell-mediated immune response, that this is a response geared towards intracellular antigens or intercellular pathogens, the intercellular bacteria, viral infections, tumor antigens, And we've seen also fungal as well as the mm, um, protozoan infections, parasitic infections in particular. And why this occurs, especially I've mentioned to you for the case of fungal and parasitic infections, is that these two um, pathogens are difficult to phagocytose. So the cell-mediated immunity would be important for just clearing the whole cell which has been infected to kill it. And I gave you also the example of the cell-mediated immune response targeted at virally infected cells, because I gave you example with the HIV virus, but you can talk about any other virus, like you talk about the herpes viruses, which occur in two phases, the latency phase as well as cytotoxic phase. So when a cell has been infected and it's in latency, the antigen is very difficult to present to the effector T cell. So cytotoxic T cell will not be recognizing it. However, signals would still be sent, which can enable the cytotoxic T cell to kill that cell which is harboring the herpes virus, which is now in its latency form. So it will clear out that cell which has been infected in future, not to allow the proliferation of that cell, which would result also into generation of the more virions of that, the more virions of that herpes virus to infect additional cells. And to that point, and then I leave you unless you have a question. Anybody with a question? Are you there?
I don't think there are any questions. Thank you for taking us through the lecture.
All right, if you're okay, then I'll send this lecture notes later. Just share with me the class email again. I don't know how we did it last time, either sending it to you and you sent it to the class. Yes, you have a question? No. Class rep, you'll share with me the email address that we used last time to share the lecture notes, and I will do it maybe within the day or tomorrow.
Okay, no problem. Thank you
Oh,
again.
have a good day.
You too.
Hi everyone, we will be using this same